Okay, so what we covered on Monday was the bare bones. We said the cell cycle is composed of interphase, mitosis, cytokinesis. These are the three parts. And there's two reasons why a cell would undergo the cell cycle. First and foremost, if you are growing, because that's a characteristic of being alive. And second is if you are repairing damaged cells. So like you break a bone or you get a cut on your skin, etc. Now, the types of cells that undergo this process are called somatic cells, S-O-M-A. Soma means body. A body cell is essentially any type of cell that consumes you. So that could be skin, that could be blood, that could be bone, that could be stomach, that could be, you name it, cardiac, right? It doesn't matter. If it's a cell on your body, it's called a somatic cell. The only cells that will not fall under the somatic cell category are going to be your gametes. So there's only two types of cells, somatic cell or a gamete. The gametes are very specific. If you are a female, you have an egg. If you are a male, you have a sperm, okay? So when we were doing the activity with your questions, it was very clear that you would pick every single answer choice except for egg and sperm. And that was on yesterday's activity, okay? Now, with the cell cycle, the one thing that the STAR test has begun to do is that it used to be taught to you like this, where we'd start at like 12 o'clock, so like right down, right, straight up and down. We would start here at 12 o'clock and then we'd work our way around the circle. Well, the STAR test has decided to like shift it over some so that way it throws you off, because kids were going to want to start here and you don't begin the cell cycle in anaphase, right? And that's where they get thrown off. To review interphase. Interphase is composed of G1, S, and G2. G1 stands for growth. S stands for synthesis. And then the first couple of weeks of school, we actually said that synthesis means to make, because we talked about organisms that were photosynthetic or chemosynthetic. And what that means is their nutrition. So if they were photosynthetic, they're making from the light. If they're chemosynthetic, they're making from chemicals. Synthesis means to make. So in the S phase, what we're actually making is these copies of DNA, because we're undergoing DNA replication. G2, we're going to finish our growth. We're going to check for error, and then we're going to move on into the division of the nucleus, which is mitosis. Mitosis is typically remembered by using PMAT. Now, I teach the entire cell cycle with hand gestures, and if we were here all in person, we usually like dance to the Macarena and do our little hand gestures once we all know them. So I'm going to teach you the hand gestures, and then you'll have about 20 to 25 minutes left at the end of class to work through today's activities. Now, you do have multiple attempts at today's activities, but please wait until I show you how to work through them, because there is some confusion there from some of my kids. Now, did it glitch again? Oh my gosh, I'm so scared that my camera is going to die. So you're going to begin with a fist. This fist is going to represent a cell. This cell is going to undergo the cell cycle because it is needed for growth. Okay, so the first step of the cell cycle is G1. G means I'm growing. So this cell is about to grow. The next step is synthesis. Synthesis means to make. What am I making? A copy of DNA. So I went from a cell, G1, grow, S, replicate. I have an exact copy. G2, I finish growth and I check for error. If everything's good to go, it's glitching, right? Oh, God. Okay, so if everything's good to go, we're going to now be ready to enter mitosis. So again, interphase, we have a cell, G1, it grows. S, it replicates. G2, it checks for error. We're good to go. In mitosis, we enter PMAT. The first step is prophase. In prophase, we are actually going to dissolve the nucleus. The reason we dissolve the nucleus is because DNA is actually stored inside the nucleus, and this is the only way that we'll be able to access it to make a copy. So if we're continuing our hand gestures, the next step is we have a cell, G1, it grows, S, it replicates, G2, it checks for error, it's good to go. Now, oh my gosh. In prophase, the nucleus dissolves. So I'm exposing the chromosomes. So you just cross your little fingers. Please don't die, camera. I need to show the little hands today. So G1, S, G2, prophase, I have little crossed fingers. The X's are your chromosomes. After prophase, we have M, metaphase. Meta pertaining to the middle. The X's are all going to have a midpoint. That midpoint is called your centromere. The centromere of your chromosome lines up against that metaphase plate, which is the middle plate. It's like an equator, if you will. Sometimes you'll hear it referred to as an equatorial plate. Sometimes you'll hear it referred to as a metaphase plate. Sometimes you'll hear it as a midline. They're all going to refer to that middle, right? So meta, always think middle. Prophase, chromosomes are exposed. Metaphase, they line up in the middle. The next letter is A, away, for anaphase. So these spindle fibers have been created. The spindle fibers are going to now play tug of war. They're going to attach to the centromeres of these duplicated chromosomes, and then they're going to begin to fish and reel them in. When they split the centromere, if you have an X is essentially two V's stacked on top of each other, right? So we had a cell, G1, it grows, S, replicates, G2 checks for error, we're good to go. Prophase, we dissolve the nucleus, expose the chromosomes. Metaphase, they line up in the middle. Anaphase, they're being pulled apart. Telophase, they're going to continue to be pulled apart, and they're going to resemble eyeballs. Okay? Now, these eyeballs are going to be the last step of mitosis. And so in prophase, our first step, we dissolve the nucleus. In our last step, we're going to have to form it once again. So the nuclei, which is plural because now we have two, is going to, it's going to be, what do you call it, created so that we can get ready to actually split the cell. Oh my gosh, camera, please don't do it. Now, every year I try to tell the kids, like, these are your binoculars. They look like minion eyes, whatever. The kids always remember it as telophase, or the boobs of the cell. And it does not help that the scientists decided to call this part right here the cleavage furrow. I don't care how you remember it. Just remember that anaphase and telophase are going to look distinctly different because you're going to have the cleavage furrow and telophase. You're still pulling them apart. And in telophase, we're actually rebuilding the nucleus. Now, in a perfect world, these are the pictures that they would show you on the star because they make it super easy for you to identify which step of the cell cycle you are in. Here I have X's. I can see the chromosomes. The nuclear membrane is dissolving. So when you think back about your little hands, we had a cell, the cell grew. The cell replicated, it checked for error. When we first introduced the little X's and we were able to see them is when we entered prophase. So this picture is actually showing you prophase. You see the chromosomes that look like X's. You see the nucleus, but it's broken apart. You see the spindle fibers beginning to form. The next step is when they all line up in the middle, that equatorial plate. The centromeres, which is the middle of the X, this is your centromere, it's in the middle center. They're going to line up, and now these spindle fibers are going to attach to them, and they're going to start to tug of war, reel them in. So prophase, metaphase, anaphase, we're pulling them apart now. Telophase, we continue to pull, but now we are seeing this cleavage furrow, and the nuclear membrane is starting to reform. Ideally, these are the types of pictures STAR would show you, but instead they're going to show you pictures like this. These are not as easy to identify. However, you know that this cell is in prophase because you can see genetic material. It's nice and dark. You can see that it has a border, but it's broken up. And those little fragments tells you that you're in prophase. Here, all of your DNA is lined up across the middle. This is your metaphase. Here, your cells are being pulled apart. Here you see a cleavage furrow. So our last step is cytokinesis. Cytokinesis literally pertains to cytoplasm kinesis movement. This is the movement of the cytoplasm that causes the cell to split. In plant cells, what is going to allow the plant to split is this formation of a cell plate via vesicles. So it actually builds like a little wall, and then it allows the cells to split that way. Now, just like we have stop lights in our roadways, you get a green light, that means you go, you get a red light, that means you stop. We have two reasons why we undergo the cell cycle. So there's two reasons for the light to be green. You're growing or you're repairing. The moment that those two stop, your light turns to red. Just like we have turns out on the road, someone is bound to pass the red light. There are cells in your body that will bypass these checkpoints, and they'll start to accumulate and create cells that are not needed. When we make cells that are not needed, we call these tumors, which we're going to lecture on tomorrow. Okay? All right, guys. We have a fire drill. So I'll post this video. You have those two activities, and I'll post a how-to on those two activities.